Okay, it gives me great pleasure to welcome Mónica Carvalho. Mónica is a Colombian botanist and paleobotanist. She obtained her BSc in biology at the University de Antioquia in Medellin in Colombia, and a master's in geosciences at Penn State University in the US, and later a PhD in botany from Cornell in the US. She joined the Smithsonian Tropical Research Institute around 2017 as an Earl Tupper postdoctoral fellow, and is currently funded by the National Science Foundation. And as you see, her work aims to understand how plants and tropical ecosystems have changed through geological time. So thank you for speaking to us, Mónica, and over to you.
Oh, thank you very much for the invitation. It's truly an honor to be here. And so, as you mentioned, I'll be talking about some of the deep time evolution of tropical rainforests. So basically, in a nutshell, what we found and what I'll be showing is how extinction at the end Cretaceous, so 66 million years ago, triggered this anomalously long period of time of low plant diversity in the tropics that actually led to the origin of modern neotropical rainforests as we know them today. So when I talk about, or when I mention neotropical rainforests, these are just new world tropics, new world lowland rainforests. And these environments overall are absolutely fundamental for Earth systems. They're not just only centers of biodiversity, plant and animal biodiversity, they're also fundamental for terrestrial carbon uptake. They have a great control over hydrological processes and regions. And these ecological services are fundamental for human health and for how Earth and biogeochemical processes operate today. So what I do and my team does is that we use the plant fossil record, so leaf floras mostly, and on the pollen record to understand the deep time evolution of these neotropical rainforests. And we do this by looking at the fossil record in tropical areas. So for the last 20 years or so, we have been studying what was the effect of the end Cretaceous on tropical floras. So we as biologists don't really tend to think much of the effect of the end Cretaceous on plants. And that's because we tend to associate this to dinosaur extinction. So there's no equivalent in plants. There's no large clade of plants that almost drastically disappeared or almost completely disappeared during this event. But we tend to forget that this was a global ecological catastrophe that ended up in extinction and was ultimately triggered by this massive 10-kilometer wide asteroid that crashed against the shallow marine platform of the Yucatan Peninsula, created a crater that's around 150 kilometers wide. And when it crashed, it instantaneously vaporized hundreds of thousands of tons of rock instantly, releasing large amounts of soot into the air, large amounts of sulfides into the air, and also all this vaporized rock kind of condensed as it was bursting out into the atmosphere, kind of condensed again into these little tiny glass particles that reheated as they were coming back into the earth and kind of created this shower of fire all around the earth. And we know this because we find these deposits of micropectites. So these are little glass spheres around a millimeter in diameter that were actually formed during that event. So they're formed by the condensation of all this vaporized rock, and they fell back down into the earth. And in this paleogeographical map, you can see this is at 66 million years ago, roughly. All these red spots are areas in which we find these types of sediments that we can trigger exactly to the rock that vaporized. And it is also associated to the iridium layer that was formed when the asteroid also vaporized as it crashed down to Earth. So what you can see here is that the effects of this debris fall was global. and probably triggered wildfires all over the earth that further contributed to this, black cloud that stayed up blocking sunlight for several decades. And from the marine record, we know that photosynthesis and primary productivity basically stopped for a few decades at the very least. So what happened to plants? That's something that we don't usually think about, but that's something that we can look at in the rock record, especially using pollen. So plant extinction has been recorded to be quite variable across the globe. So ranging from around 30% extinction rate, and this is talking about regional extinction in a particular area, such as in the Great Plains in the United States, and down to only about 10% extinction rate. So most plants that lived in Patagonia and Antarctica were able to actually come back. But we don't know really why there's such a difference. It could be related. One idea is that it could be related to distance, just plain distance to the impact site. But one thing that is quite clear is that most of these studies show what happened in high temperate areas. There's actually very little, almost no information of what happened to plants during this event in tropical regions. And the reason for this, and I'm pretty sure I don't have to preach to a choir, is because the tropics are great for looking at modern plant diversity and modern plant biology, but they're absolutely terrible for looking for fossils. And this is because to look for fossils, we need access to fresh rock. And most of the tropics are either covered by lush vegetation, and the soil cover is usually pretty deep. And that is actually very difficult. That limits paleontological findings in tropical regions. What our team did was to focus on two different rocks that could tell us what happened exactly 66 million years ago. So the first approach was to use pollen. And the pollen fossil record is great to address these questions of extinction and diversity through time. And that's because pollen is very abundant in the rock record. So we can actually collect little fragments of rock, isolate pollen from there, and we get a lot of, we get a lot of pollen data. Pollen is very, very abundant. And because it's so easy to take out of the rock, we can very easily have a nearly continuous record of how these pollen communities looked like through time. When I talk about pollen, I also mean pollen and spores, by the way. And usually these records, they reflect regional vegetation, so community structures at a regional level. And they give very good detail of that diversity through time, as I was mentioning. So what we did was to build a composite record that would span from 72 to 56 million years ago. So that is the whole of the Maastrichtian, which is the last period of the Cretaceous, and the whole of the Paleocene, in order to have an idea of how diversity changed through that time and what was the extent of the extinction back then. We did this by sampling 39 different sites. So these sites were either cores that had been drilled by oil companies, or sites that had exposed sections. So each one of these sections has varies a little bit in age, and we were able to find or build this composite record spanning the extinction events. We counted or our team counted over 50,000 individual grains, either spores or pollen, in order to build a very robust data set that could tell us about extinction at a regional level. This we complemented by looking at fossil leaves. And the reason that fossil leaves complement very well the pollen record is because they're reflecting local vegetation. So usually these fossil leaf deposits are actually formed in channel or floodplain deposits, which means that they're effectively recording kind of moments, individual moments in time. either of the leaf litter that is rapidly buried in some flooding event, or in channels, which are ultimately reflecting nearby vegetation that is kind of transported and rapidly buried in order for it to become fossilized. So we looked at 3 different floras. One of them was a pre-extinction flora, which is the Guadalus flora. It dates from about 70 to 66 million years ago, so that's late Cretaceous, and 2 post-extinction floras, one in the Cerrejón Formation, which is in northern part of Colombia, and the Bogota Formation, which is actually, it crops out near Bogota, Colombia's capital. And these 2 post-extinction floras are dated to be Paleocene, so around 60 to 58 million years ago. And overall, in order to have a good representation, we looked at over 6,000 fossils. Some of these fossils were collected from census sites, and these census sites are places in which, from a definite volume of rock, we collect and identify all the leaves that we can possibly find in order to have a comparable data set that we can compare and can quantify and compare with either modern flora or with other fossil floras. And so these leaf fossils we find, as I was mentioning, because it's so hard to find outcrops in the tropics, what we do is that we rely on mining efforts. So either coal mines or siltstone mines that have previously removed all the vegetation cover, all the topsoil cover, and they really expose vast quantities of rock. And in these rocks is where we can actually find our fossils. So these mining efforts can be either huge coal mines, or, such in this case, kind of small mining efforts. And this siltstone mine in particular is found just outside of the city of Bogota. And in these places, we've been going over the years, and we basically collect with rock hammers and pickaxes. We extract large pieces of rock, we split them open, and inside the rocks is where we find our beautifully preserved fossils. And we find flowers, we find fruits, and we find leaves. Leaves are the most common, and because they're the most common, they're most abundant, they are the ones we rely on mostly for kind of these ecological inferences and statistical comparisons. So what did we find? Is that diversity throughout this Maastrichtian and Paleocene period changed quite a bit. And we see that there was a drastic decrease in plant diversity just right around 66 million years ago that is correlated with a significant increase in extinction rates and no changes in origination rates. And this, because there are no changes in origination rates, we can actually say that this drop in diversity is mainly to do to extinction. We also saw that it took about 7 million years for plant diversity to reach pre-extinction levels. So this is quite a pretty long of low plant diversity in the neotropics, which is absolutely strange, since currently we see that these are centers of biodiversity. And associated to this extinction, we see that there's a general differentiation in plant communities. So when we group these plant communities, these spore and pollen communities, we see that there are 2 distinct clusters. So all the plant communities that we find in the Maastrichtian, they group together. They're more similar to each other. And when we go into the Paleocene, we find that there is another different group. This is telling us that there's a complete community turnover across the extinction event. And this community turnover has a lot to do with a drastic change observed in the proportion of flowering plants. So before the extinction, all these Maastrichtian communities were composed of roughly 45 to 50% flowering plants. After the extinction, the proportion of flowering plants that we find in these pollen assemblages reaches up to 90% and becomes quite steady. This is roughly the same percentage that we find, if if we go to a tropical rainforest today, we isolate pollen and look at plant community composition. So what did the leaves tell us? So leaves, as I mentioned, they're a great complement to this overall picture of change in diversity and change in plant community. And that's because they reflect a lot of the ecology of a forest. So they reflect both biotic and abiotic conditions that ultimately end up translating either into leaf traits or leaf characteristics that we can observe in the fossils. So we can look at things such as precipitation and temperature, and this is because these climatic factors influence leaf physiognomy. So leaf shape, leaf size, leaf margins. We can look at canopy structure, and that's due to the effect that we see on the isotopic signatures of leaves. We can look at plant composition, specific plant composition, and that's because it's a lot easier to identify leaves than to identify pollen grains. With pollen grains, especially pollen grains that are so old, it's quite hard to get even to a family level when we're looking at them. And we can also look at plant-insect interactions such as herbivory, which are ultimately reflecting an ecological process that happened millions of years ago. So what we found when we compared these post-extinction to pre-extinction floras is that overall, these three different sites, they represent or they're consistent with tropical rainforest environments. So the size of leaves, most leaves are quite large. Most leaves have untoothed margins. There's a lot of abundance of drip tips among our fossils, which ultimately is showing that we have tropical rainforest-like environments, which is also consistent with temperature and warm temperatures and the rainy conditions that we know prevailed across the Maastrichtian and the Paleocene. We see how across is because of the effect of closed canopies on
Mónica, just to say, Mónica, so we're occasionally losing sound, so perhaps it's best to switch off your video and see if that just stabilizes things better. There.
Okay. There.
Okay, let's try that. Thank you.
Okay, so one of the reasons, or the main reason we can identify these closed canopies or differentiate closed versus open canopies in the fossil record has to do with that isotopic signature that is different between understory and canopy leaves in closed canopy forests. And this is the closed canopy effect. And that effect is mainly due to the restricted atmospheric mixing. So the air that is trapped in the understory doesn't really mix much with the air that is above the canopy. Soil respiration depletes CO2 in the understory. And that added to the fact that there are differences in photosynthetic rates and humidity rates across the forest canopy, ends up translating in this very drastic, nearly around 8 per mil difference in the isotopic signature of leaves in the understory versus leaves in the upper canopy. And what is really cool about this is that we can actually see these differences if we look at leaf litter, at modern leaf litter, or if we look at fossil leaves. And that's what we looked at. So in this plot, you can see these bars. They represent forest litter. So it indicates that, as we sample a larger number of leaves, we are actually able to capture that range, that large range of isotopic signatures that is typical of a closed canopy forest. When we look at our fossil sites, the Cerrejón site, this one is a floodplain deposit, and therefore it reflects kind of the leaf litter that existed back in the Paleocene. We can see that the range of delta C13 is nearly the same as we see in closed canopy forests, whereas our Maastrichtian site, so pre-extinction flora, does not have that range of delta C13s that we exhibit. So we interpret this to be more of an open canopy forest in which there is not that there is a lot more mixing. So that effect, that closed canopy effect, is not present in the pre-extinction flora. So one thing we've been doing with these fossils over the years is also identifying them. So doing taxonomic revisions and placing them phylogenetically. And we've been able to see very distinct plant families, not just a difference in the proportion of flowering plants, but also what families of flowering plants we find either before or after the extinction. So after the extinction, we've been able to recognize some very typical plant families, tropical plant families that we see today. We've been able to identify members of Cormalvoides in the Malvaceae. We've been able to identify, we find melastomes, we find Menispermaceae, both as leaf compressions and various types of endocarps, indicating that there's quite a lot of diversity of this family back then. We've described several members of the Oraceae, various Ericaceae, different species, different lineages. This one over here is something that relates to Cocos and Nipa. And the most striking thing is definitely the abundance and diversity of legumes that we find in these post-extinction rainforests. We have, in these two floras, we have 5 different types of legume leaves, which ultimately could represent species or different genera, and 8 different types of fruits, which is actually showing us that legumes back then were not only abundant, they were also very diverse in these ecosystems that originated out. Before the extinction, we don't really recognize a lot of the families, a lot of the tropical families that we see today. The main reason is because as we go further back in time, leaves and reproductive structures become a lot harder to identify. And we see that there's a lot of homoplasy in the leaf traits that we observe among these fossils. We have been able to identify, however, in the pre-extinction floras, members of the Rhamnaceae, those as reproductive and leaf structures, and members of Viperaceae. So one thing we can do, because we have these fossil census sites, we can compare them with modern vegetation. Specifically, we can compare them with the plant family composition that we observe in neotropical rainforests today. The reason we can do this is because it's due to previous taphonomical studies. So taphonomy is the science that kind of studies all these processes that lead from something that's living to how it becomes fossilized. So previous studies show that litter samples in tropical rainforests that are analogous to our fossil sites, they reflect up to 90% of the standing biomass in a 12.5 meter radius, which means that if we take a litter sample, we can actually see, we'll have a good representation of most of the plant diversity, or most of the plants that are there in a 12.5 meter radius. So what we did was take this same idea and compare our fossil sites to 2 neotropical rainforests. So permanent plots in Barra Colorado Island in Panama and Parque Macajaco in Colombia. And these permanent plots, they're great. Everybody here knows, every single tree is identified. And what we did was kind of randomly subsample different areas and compare the plant family composition across these different areas to create kind of a null distribution to see how different is the forest within itself. And we also compared the plant family composition in these little areas to the plant family composition in our fossil sites. So basically we were asking, are the angiosperm families that we find, is the composition of angiosperm families that we find in the Cretaceous different to today, and is the one that we find in the Paleocene different to today? And what we find is that consistently, regardless of the plot size we selected, so the plot size is this, we see that the Paleocene rainforests, so post-extinction rainforests, are much more similar to modern neotropical rainforests than those of the Maastrichtian. And also, these two are different between each other. So the composition, these Paleocene rainforests were not just, they didn't have a greater abundance of angiosperms. They also had different angiosperms. The composition of plant families was different across the extinction. Another thing that we looked at was leaf herbivory. And this is actually something that's really cool in the fossil record. And that's because, as insects are feeding, they make these little trace marks. And these trace marks, they're very nicely preserved as fossils, and we can identify them from fossils due to this distinct reaction rim. I mean, not just because of the morphology of insect mouth parts, but because, when insects are eating, they induce this kind of reaction tissue in the leaf that we can identify in the fossils. And this is actually a very powerful tool when we look at forest ecology in the past. And that's because, insects have many different ways in which they can eat. These can be like very generalized ways, and they're just chewing from the margin, they're just making holes. But then we have also these kind of very highly specialized associations, such as leaf miners and gallers. And in the fossil record, what we can do is that we can reduce all of this variation of different ways of feeding into discrete trace fossils. And these discrete trace fossils that reflect functional feeding groups can ultimately serve as a way to quantify and compare herbivory across different sites or even across different plant hosts. So what we did, or what we found in our fossils, is that consistent with what we expect in tropical floras, herbivory was very high. So nearly 50% of all our leaf fossils, both before and after the extinction, had chewing marks. But the distribution of these chewing marks across host plants was very different. So what we did was kind of compare how similar were these different chewing marks across plant host pairs. And what we find is that in the Paleocene flora, so after the extinction, there is a very high similarity. This is a dissimilarity index, so that's why it's inverse. There's a very high similarity of leaf damage across host pairs. So basically that means that almost all plants had the same types of chewing marks. Whereas in the Guaduas flora, so in the pre-extinction flora, we see that there is very low similarity. So that means that distinct different host plants had very different types of chewing marks. And because these chewing marks are ultimately reflecting the insect communities, we can infer that there was some degree of host specificity among the insect communities when they were feeding before the extinction. And after the extinction, we have a completely new type of insect communities that were behaving very differently, and they were just basically chewing everything that was around. Put together, the pollen data, what the leaves are telling us about the ecosystem, we see that, both before and after the extinction, we have rainforests that complies with, our understanding, our geological understanding of climate across the Cretaceous and the Paleocene. But we see that before the extinction, we had these mixed communities that were made up of ferns, conifers, and flowering plants. So once again, flowering plants were only about half of what we find in the pollen and spore assemblages. They had open canopies, and they had very host specialized herbivory. Whereas the post-extinction rainforests, they became fully dominated by angiosperms. And this difference kind of triggers some fundamental distinctions in transpiration capacity and productivity of rainforests. And this is associated to how flowering plants work. These rainforests were also forming closed canopies. And this is telling us that there is a drastically increased vertical complexity in the forest. We also see that there are legumes and other tropical tree families that are typical of rainforests today, and herbivory was widespread. So basically what we're seeing are these two very different types of ecosystems that function differently. And that these ecological services, for example, that we recognize today from tropical rainforests are due to the abundance and dominance of angiosperms, of broadleaves in general, their transpiration capacity, their productivity, the number of leaves that we find per forest strata. Those characteristics only came to be after the extinction. So it is quite possible that it was that extinction event that pronounced decrease in 45% of plant diversity that ultimately led to this reconfiguration of the forest and kind of the origin of a new type of forest. Well, the question is why? Why was there such a profound change in ecology and taxonomy across the end-Cretaceous? It's not, and the real question there is, well, flowering plants by the latest Cretaceous, they were already pretty diverse, not only taxonomically, but also ecologically. So we had trees, we know that there were trees, we know that there were pretty abundant, we know that there were shrubs, herbs, aquatic plants. Why, it's not like they would not be able to form these closed canopies. And also, why did the forest structure and composition change so much? Why didn't it return to how it was before? So aside from the intrinsic things that flowering plants have, kind of these ecological advantages that they have, we can kind of at an ecosystem level that could have contributed to this. And the first one has to do with disturbance. And an analog to this is the effect that large herbivores have on their surroundings. And we see this in the African savanna with elephants, and we see it in Pleistocene Park with the reintroduction of these large herbivores, and how the reintroduction of these large animals is actually shaping the type of forest that you find, kind of cutting back the forest and making more open environments. And this is something that could have been happening back in the day. So even though we don't have a very complete record of what the dinosaur communities were in the northern part of South America, we do have an idea that at least some really large animals lived there. So from 100 million years ago, there's the Padillasaurus, which is found in Colombia, has an estimated weight of 10 tons. Amazonsaurus, found in the northern part of Brazil, weighed about 5 tons. So these were pretty large animals. And even though it's just such a small fraction of what could have been living there, we already know that there were some large animals there. So it could be that back in the Cretaceous, these animals were actually disturbing the forest in such a way that they kept the canopies open, knocking down trees, moving around the forest. And once they were gone after the extinction, it kind of enabled flowering plants just to express themselves and forest communities to reach that open canopy structure. Another possibility has to do with selective extinction, and that's the selective extinction of conifers in particular in the Neotropics. So conifers did not go extinct worldwide, but regionally they did. So the abundance of conifers, of Araucariaceae, which is what we mostly found in the Maastrichtian of northern South America, they were completely gone by the Paleocene. So it could be, and it's typical that, when we look at the fossil record and how extinctions have hit, usually those lineages that have a narrow ecological range, they're more severely affected than lineages that have a broader ecological range. So it could be that, Araucariaceae, we extrapolate a little bit from what we know about them today. They're mainly large trees. They're all doing kind of the same. there could have been selective extinction against them, and then once they became regionally extinct, they kind of opened up that niche, that upper canopy niche for flowering plants to fill up. And our third hypothesis has to do with an initial soil fertilization effect that could have been operating because of the impact of the asteroid. And the reason we think that is that overall we know that conifers, they grow better in poor soils than flowering plants do. Flowering plants overall, they grow a lot better. They're benefited when there's high nutrients. They grow a lot faster. And during the Maastrichtian, the Andes were just beginning to uplift. So there's still quite of a debate of whether there was, significant topography and a significant kind of, minerals being eroded and contributing to these poor soils that were found in the Maastrichtian. So it could be that conifers were living okay because they were growing in poor soils. And then one of the things that could have happened with the impact of the asteroid was that there was this, when we think about the abundance, the sheer abundance of soot that was ejected into the atmosphere, all the ashfall that was related to wildfires, all the tsunami debris that is associated to the impact, that could have led to an initial enrichment of the soil. So kind of an initial fertilization that could have helped give a jump start to flowering plants, just to start taking over. Then further, as we see that the abundance and diversity of legumes increases in the Paleocene, and because they're overall related to nitrogen fixing bacteria, this could have further led to an enhancement of the nutrient cycling, therefore kind of given a positive feedback for the abundance of flowering plants and the domination of flowering plants in tropical rainforests. So all in all, one of the things that we ultimately found that is quite relevant to what's happening today was the proportion or the extinction rate that we found back in the Cretaceous. And we found that around 45% of tropical plant species went extinct. And this number is funny or curious because current projections of extinction rates of Amazonian plants are right around those same rates. So with current deforestation rates, by 2050, it's expected that around 50% of Amazonian plant species or tree species will be endangered. So it's something just to think about. When we look at this fossil record, we see extinction that occurs in a geological instant, and it's absolutely terrifying to think that these same type of extinction that we see when an asteroid hits the Earth is kind of what we're expecting due to habitat destruction. So that's just something to think about. And with that, I thank you very much. And if you have any questions, I'll be happy to take them.
Thank you. That was terrific, Mónica. If you want to switch on your camera again, and
yes,
that's hopefully it'll be good. Great. Terrific. A very, very nice, clear exposition as well as some fantastic science. Okay, we can open up to questions now. Again, I encourage people to have their cameras on and either pop your question in the chat or directly raise your hand. And if you're able to, I'll encourage you to directly ask Mónica your questions. While we're waiting, I'll just kick off with one, which is of your three hypotheses for why the angiosperm began to dominate afterwards. And one is around selective extinction, that the lowland conifers disappear from the neotropics. However, if we look at Southeast Asia and Australia in particular, we've still got tropical Arocaceae like the kauri in tropical forest biomes. So that would so you didn't get local extinction there. So if all that was required was local extinction, then you'd expect sort of a Maastrichtian type forest to persist to modern times in Southeast Asia and Australia. And the fact that it doesn't perhaps argues against that hypothesis, and more the other two, that the extinction of dinosaurs or this nutrient pulse were the factors that dominated there. I wonder if you have any comments.
Yeah, no, that's absolutely right, and that's one of the, I think, like, we personally feel it's fantastic about Southeast Asian forests. The fact that we still find Kari pines and even lowland Araucaria, and it also, to the one way I interpret it is has to do with maybe even extinction was not so severe over there. So that's maybe one reason why conifers may have kind of persisted there. Although it's that area of the world has a very weird geological history. So back in the Maastrichtian, Southeast Asia was underwater. So kind of all this flora that we have there is kind of like recolonized, and diversification and radiations that have occurred more recently. But to me, it's also indicative that that side of the world was not so severely hit. That's my interpretation, though. And it's definitely true that, and that's why we propose these three hypotheses, not even as mutually exclusive. These are just factors that could have contributed to what we see happened in the neotropics. But that's completely, it's a very, very good point. We still see forests in which there are tropical rainforests, lowland, in which we see araucarias, in which we see podocarpaceae even, but they're not a dominant thing. They're not a dominant feature as they could have perhaps been back in the Maastrichtian.
Thank you. Anybody else for a question? Lucia?
Thank you. Well, first of all, thank you so much, Mónica. This is wonderful, really, really wonderful presentation. And so, well, one thing, first of all, is I look at the history of Amazonian forests from a phylogenetic perspective. And what you showed is perfectly consistent with everything we've been finding in the past. So in some ways, it's such a relief to hear your talk and see that all the pieces of the puzzle actually match. And so we look at, phylogenetic data of insects, of birds and plants, and we see exactly the same pattern, just, extinction and lots of diversification happening after the extinction. So it's really, really wonderful to see all the pollen and the leaf data, leaf fossil data, and just seeing data from different areas corroborating the same hypotheses. One question I had was about the herbivory, and it actually cut a little bit, so I couldn't follow your argument completely. Because you mentioned that, so if I understood correctly, that so pre-extinction, there was low similarity in the chewing marks, and post-extinction, high similarity of the chewing marks, right? And yep. So, and I was just thinking if that could be actually indicative, if there's low similarity, if that wouldn't indicate higher insect diversity actually before, which would be exactly the opposite of what we would expect, because we would expect higher afterwards. But I just wanted to hear your thoughts on that. And maybe you can recapitulate also your line of thought about the host specialization associated to that.
Yeah, okay, so overall, there is a general correlation between, the numbers, the diversity of chewing marks and the diversity of culprits. So if you find a tree that has, a high diversity of these different chewing marks, it's quite likely that there's a high diversity of insects that need it. When we look at the, at the diversity of these chewing marks across the extinction, we see that it's not a very consistent pattern. So there's high diversity both in the pre-extinction forests and in the Bogota flora, which is one of the post-extinction ones. So just by the numbers, just by different types of damage, we're not really able to say whether there were more or less insect species, herbivore species. But what we do see is this distribution pattern of chewing marks. So because the chewing marks in the pre-extinction floras are very different across the host plants, we can say that it wasn't the same species of insects feeding on these two different plant species. There were different insects that were eating these different plants. That could be related to a greater diversity before, but we don't have really a way to test that. What we see afterwards, after the extinction, is this pattern in which it's basically all leaves, all plant species are eaten the same way. We cannot really say that it were just generalized or generalistic insects that were feeding on everything, but we do see that there is a change in the behavior of the insect communities. So that behavior could have changed or likely changed because the insect communities per se changed, which is also consistent with with an extinction. The plant hosts go extinct, so if there are insects that are host specific, they will also go extinct. And then we have these new niche, new opportunities, new niches open for insects to eat anything that they can basically find. But it's hard to say anything about diversity at this point, especially because the numbers of different damage types are roughly the same between the Guaduas flora and the Bogota flora. I don't know, did I answer a question or did I make it more confusing?
Yes, thank you.
Okay, thank you. Any more questions, anyone? Chad?
Yeah, thank you. Yeah, I would just reiterate that was an amazing talk. Your work is just fantastic, and I think it's actually some of the most exciting stuff for me to follow anywhere in the world. It's just really, really cool stuff, and I'm really excited to have gotten to hear about it today. So I was wondering, though, kind of related to Yadvinder's question. So the angiosperms had this explosive diversification in the Cretaceous, but it occurs to me that maybe that wouldn't have necessarily corresponded with a rise of numerical dominance, like numerically taking over from pteridophytes and conifers, and that maybe would have taken longer than the phylogenetic diversification. So I guess maybe almost as a null model, alternative to some of the hypotheses you presented, could it be that angiosperms were going to replace gymnosperms and pteridophytes anyway, and so the meteor impacts wouldn't have necessarily directly driven that takeover, but instead maybe just altered the pace? I'm curious about your thoughts on that.
Yeah, so there's something that's very interesting with that increase in angiosperm abundance is that it's not only something we observe in the tropics. It's also something that when you look at the fossil record in New Zealand, it also happens. When you look at it in North America, it also happens. So it's like we see this common pattern in which angiosperms, after the extinction, angiosperms just took over. What you mentioned about that initial diversification of flowering plants is true. So by the late Cretaceous, flowering plants were already really diverse, and this kind of goes back to Darwin's abominable mystery. It's in the fossil record, we find that these late Cretaceous floras, they're full of flowering of angiosperm leaves. So what Darwin referred to was dicot leaves, so eudicots and basal angiosperms. But the fact that they're there and that they're functionally diverse doesn't really mean that numerically, as you said, they were dominating the ecosystem. So one thing is that phylogenetically, ecologically, they were diverse. The other is that they were actually dominating ecologically, as they do today. Kind of as a landscape. it could be that that they were, slowly displacing ferns and conifers. But why hadn't they done it before? I mean, it's why by 66 million years ago hadn't they fully displaced conifers? And that's kind of the key question. So one of the when as paleobotanists, when we got into looking into what happened in the end-Cretaceous, we always ran back into this idea of when did rainforests originate, tropical rainforests originate. And because the fossil record, we find a lot of overall fossil leaves, fossil angiosperm leaves, there was this idea that tropical rainforests existed almost since flowering plants diversified. As a biome, that's not true. And one of the things that what we find is that as a biome, this sort of this that we find today in neotropical rainforests, it only appeared after the extinction. And we're talking about flowering plants started diversifying around 120 million years ago, and the extinction was at 66 million years ago. It could be that a null hypothesis is that flowering plants were already displacing, but why hadn't they fully displaced ferns and conifers? I mean, that's something that we will absolutely never know unless we have some sort of time machine. But it's definitely an interesting thing to think about, especially because we live in an angiosperm dominated world, and we think of angiosperms as they just took over and they dominate everything. But maybe if there hadn't been this mass extinction, we would still live in a world that's equally mixed between flowering plants and ferns and conifers.
Yeah. Just to add a perspective on that, if we consider a modern African savanna where we have dicots and monocots existing in an equilibrium mediated by megafauna and fires, we don't say that the dicots are taking over the monocots. If it wasn't for the megafauna, it's a dynamic ecosystem that's being created that's equally parts controlled by the large animals and the different plant communities in competition. And I wonder what we had in the late Cretaceous was a similar system. Normally we think that once you go away from savannas, you get into a forest, the megafauna aren't large enough to have a strong effect on structure. They're not big enough to knock over trees and to suppress things. So the forests are bottom-up controlled rather than animal controlled. But maybe once we have these super megafauna of the Cretaceous, we're at a state, yes, where these sauropods could be knocking over trees, suppressing large angiosperms. So you get a sort of analogy of a modern savanna, but actually in the forest biome with these super megafauna creating a new an equilibrium system that isn't going in any one direction or another on evolutionary time. It's an end point in itself that's mediated by large animals. And then where the large animals get taken out, it flips into a different ecosystem, a closed canopy system.
Yeah, that's one of the fun thing about paleontology is that when you start thinking about the whys, that's where you start realizing that our world has been many different worlds in the past. So that's where we have to start thinking about all these different aspects, and it was actually a very fun exercise with several of my co-authors. We were just bouncing ideas back and forth of all the different ways in which these forests could have been functioning differently. And definitely, the effect of large animals, we cannot say that they were not doing anything. The same idea that carbon dioxide levels back then were higher than they are today, they also imply that there's a fundamental difference in what productivity could have been, even if there's a difference in the proportion of flowering plants versus others. And that's where it gets interesting. So it was a completely different world. We can do it, and I do think that it's quite an analogous system as I see it. I do think it's quite an analogous system to what happens in the African savanna. And at least that's kind of the closest we can get of anything today. But it was a completely different world. And it's actually telling us a lot about, how plants evolved, how ecosystems evolved, and how ecosystems have been changing over time.
Okay, any more questions?
Can I ask another one?
Go ahead.
So yeah, you're talking about the African savannas, and I was also thinking about the structure of these forests, and one important component are the lianas. So when we compare forests from Asia, Africa, and Latin America, and I was wondering if you saw anything about, in terms of, well, the pollen, and if you could say something about the structure of lianas. And I can't help it but ask if you specifically saw Bignoniaceae pollen, and I often talk to Carlos about that. So if you can say a couple words about that, I'd appreciate.
Yeah, so the evidence for lianas is indirect. So we have a lot of Menispermaceae, and we have been able to identify in the Paleocene, in the post-extinction rainforest, we have been able to identify lineages that today they are lianas. So they were probably lianas back then. We have something that looks like a very basal Passifloraceae. You see some seeds, so it's probably back there too. And in the pre-Cretaceous, in the pre-extinction forests, we see that there's an epiphytic fern, and we don't really see a lot of evidence of like angiosperm lianas. That's something that it's a lot easier to identify them if we had the wood preserved, obviously, but yeah, unfortunately we don't. And if you have some any sort of idea of leaf traits that could be strictly associated to lianas, it would be great to actually try to tease that out, because that's the only information we get from leaves. Anything that's ecological or that we can relate to a taxonomic group in particular. And unfortunately, I do know that, yeah, it's almost impossible to identify the pollen back then to family level, aside from general, Bombacoideae or palms. So yeah, we need to get better at identifying pollen and comparing it to modern families, in order to know a little bit more about that.
Right.
Super.
I see, Catalina, you have a question in chat. Would you like to ask that directly, or would you ask me?
Yeah, thank you. Mónica, it's always a pleasure to listen to you. It's fantastic to listen to this information again and again. I could stay here for hours, but I have about delta C13 that you get. I was surprised that you interpret the change in carbon isotopes just to infer like the depth and the complexity of the canopy. Did you consider like other factors when trying to deal with this data? Because the first things that comes to my mind is like climate, or even like the plant composition itself could be one factor behind the differences you got. So could you elaborate a bit more about the delta C13 data that you got?
Yeah, so our delta 13C we got from single horizons, so floodplain deposits. So because of how these things form, they're just representing kind of like a snapshot in time. So the equivalent is you have a flooding event in the forest, kind of takes all the leaf litter and it buries them, and that's where we got we sampled our leaf cuticles. So we're kind of minimizing the time span of what that litter was representing. And we also looked not at the values per se of delta C13, but we looked at the range. So a lot of these plants, these leaves that we sampled, were actually the same morphotype or morphospecies. So they were probably representing different individuals that were growing in the same forest and of the same taxon. And because we're looking at, the range that we see, so we would find values that would go from, minus 31 to minus 22 or to minus 20. We were looking at that variation. And because of our sampling, that variation is actually reflecting, it's reflecting the climate that was occurring at that time. It's reflecting the plant composition that was occurring at that time. But because we're resampling from the same species, we're kind of narrowing down to, what the forest structure was right there. So it's kind of equivalent to if you go to a forest today, you take a bunch of leaf litter and, do the isotopic analyses. The biggest signal that you're going to be finding there has to do with those, with that ecological gradient that you find across the forest canopy.
Okay, thank you. Any more questions?
Can I ask a final one?
Go ahead, Lucia. Okay.
This is so exciting. I have so many things in my mind. And by the way, Mónica, it's such a pleasure to meet you, even though it's virtually. I've heard about you through Carlos for so many years, and it's a true pleasure. And my question was just, so what's next? So what are the next steps now, and what are you planning? And are you looking at different, communities?
Oh, wow. Yeah, no, there's actually a lot of work. So one of the things I really want to tackle is the effect of legumes. So we're really trying to test whether, this increase in abundance and diversity of legumes in the Paleocene changed the nitrogen cycle. So just because, the abundance of this family and sheer dominance in modern ecosystems. So that's one thing that we're trying to tackle, both geochemically and by looking at kind of functional diversity of legumes back in the Paleocene. And we're also looking at other Cretaceous floras in Colombia. So we recently found a flora that's 100 million years old. So it's very exciting because, it's going further back in time, and we do see that there's, you find these teeny tiny flowering plant leaves and a bunch of conifers, like podocarpaceae, ginkgo-like things, and ferns. So that works completely different. So that's something I really want to keep on working, but I'm in an early career stage, so looking for jobs, and that's the first thing I have to do.
That is super exciting, and I'm already looking forward to that. I remember the early paper, like a couple decades ago, by Robin Burnham and Alan Graham, and they show the fossil record, and it's just like an empty thing throughout South America. So it's so exciting to see all this data coming up, and I can't wait to see the results from these studies. The study with legumes too sounds super exciting. So thank you so much.
Great.
No, thank you.
Thank you. Well, just as a slight follow-on, this is a question you may not be able to answer, but it will be wonderful if there were similar studies going on in the paleotropics in Africa or in Southeast Asia to see the story. Is that possible? Are there sites and deposits that are identified where a similar study could be done in the paleotropics?
It's really hard. There are some places in like off the coast of Africa that probably have a Cretaceous-Paleocene sequence in which you can extract pollen and look at pollen, but we're not really aware. We don't really know of Cretaceous floras in Africa. In Southeast Asia, we have the Myanmar amber. There are leaves there, but that's also like 98, 100 million years old, although there's a lot of debate. So that particular point in time, it's going to be hard to find elsewhere, but it would be super interesting to see, to what extent was extinction different? Was the Neotropics hit harder because we were, just so close to the impact site? Or is it something that, because of atmospheric dynamics, the tropics are a lot more sensitive to these atmospheric perturbations? Yeah, there's still a lot to do, a lot of questions.
Yeah. Well, Well, I think it's absolutely, as I think Jed said, it's an absolutely fascinating frontier of ecological and paleoecological research. So congratulations, and not only for fantastic science, but as others said, a really wonderfully clear exposition and explanation and introduction to what you did and what you found. So we have a tradition of these seminars for everyone at the end to unmute, and so we can give an audible round of applause to the speaker. So I'd encourage you to switch on your microphone.
Thank you very much for joining in.
Great. And we'll post this lecture on YouTube, so feel free to share it with your students and colleagues and later on as well. So thank you again, Mónica. Thank you again, everyone, for attending.
Thank you, everyone. Thank you, Mónica.
Have a wonderful weekend.
Okay, thank you.
Thank you very much.
Hello, everyone. Bye-bye.
Bye.